Hi, everybody, and welcome to our lecture today, which focuses on epigenetics. Epigenetics is one of the new concepts and methods of studies of the genetic information, uh, and it provides some very interesting viewpoints on how our genes are passed down and our phenotype or the characteristics that are visible on us will be developed. Here's are some of the learning outcomes that you guys can glance through. But again, the focus for us would be looking at some of the essential components related to epigenetics. So let's go ahead and start by looking at our genome. So again, if you guys recall the word genome, genome meant that you're talking about is basically all of the genes in your body that eventually encodes for a protein. Now, there's a good example that says that basically if you look at an orchestra, you have a number of instruments, and all of those instruments basically play at different intensities. Some of them might be um, throughout the whole orchestra, some of them may go quiet, some of them at the end, some at the beginning, etc. So we can relate that to our genes and how they are expressed. So not every gene is expressed at every second of every day for that cell's life. And the amount that you're producing, the amount of a gene expression, or basically the amount of protein that you create from the gene is also not always the same. So one of the first thing that I want you guys to understand is regardless of what type of cells we're talking about, all of the cells in our body have a set of genes that are called housekeeping genes. And these are basically the ones that are always going to be expressed, and they're necessary for the health of the cell itself. So there is no specialization in terms of the cell type, rather than if you are a cell, you need to accomplish certain tasks, therefore certain gene needs to be expressed. For instance, if you want to think about it, things that are associated with ATP production, things associated with hydrolysis, things associated with breakdown of glucose, things associated with process called glycolysis, etc. So again, these are essential for the survivability of the cell. Now, as we go into each category of the cell, then we kind of dive into genes being specialized. So certain genes might be overexpressed in type of cells, while they're underexpressed in another one. So those differentiations that you see basically arrives from what type of cells we are talking about. Now, what is epigenetics? So before we're talking about maybe a decade, 2 decades ago, when you looked at the genetics, they thought that the genetic information is locked. And basically, there wouldn't be any change. So that kind of like, you're locked, this is your gene, you got it from your parents, and you're done. There is nothing going to be changed. But in recent years, we're kind of noting that we have the concept of the environment and what we do and how we behave, and our cultures, what we eat, how we grow up as a child, all of these also have a role in what genes and what level of expression we will have. So when we talk about epigenetics, we're literally studying how the behavior and the environment can cause a change or affect the way the genes are working. So again, looking at this picture, they're kind of showing you, playing with the toys, books, as well as some of the, again, special things, food that you eat, your environment that you're growing, the chemicals you're exposed to. All of these can literally lead to the genes being expressed at different level. An example that I provided here, if you guys kind of read these paragraphs, you notice that it said even when you're looking at a pair of twins, even though they're identical in terms of genetic material, they may exhibit different behavior, skills, health, and achievements. So just because they share the identical genome, that does not translate to having the same identical behavior. That being said, yes, there are things that are going to be the same because they share a lot of factors, but there are also components that are going to be changed based on the individual and the concept of epigenetics. So again, it affects how the body reads the sequence of DNA. And by the means of how it reads the DNA sequence, it basically is it going to allow that gene to be expressed at a higher level or lower level? Uh, um, is it easily accessible through the process of transcription, translation, etc.? So let's kind of go and expand on this. So before, if we were looking at, again, kind of the old concept of genetics, pre-epigenetics, basically, we would say you received your genetics, there's a specific DNA sequence that controls the genetic regulation or the expression of the gene, and those genes give you your phenotype, which basically means your characteristics, who you are, like your eye color, your hair color, let's say your disposition to disease, treatment, etc., right? So before we would just say that's based on the DNA sequence. Now we say things that you eat, your environments, right? How polluted the environment is or how clean it is, your experiences as a child or, growing in a healthy family, etc., etc., can actually modify the expression of those genes and change the regulation. Regulation implying how often a certain gene is going to be expressed and therefore affecting the phenotype. Now, one of the organs that I want to talk about briefly is an organ called pancreas. If you don't know, if we kind of look at your abdominal cavity, so here is the neck. Sorry about my drawing. Here is the muscle diaphragm. And right here would be your stomach. No, it's not a shoe, it's your stomach. And then underneath, tucked underneath it, you have this little tadpole-shaped organ that's called the pancreas. So you have to lift up the stomach and pancreas is kind of tucked in underneath it. Now, pancreas is a very important organ for us because it's considered a dual gland. Dual gland implying that it has basically two functions. One of the function is identified as exocrine versus the other one is endocrine. Now I'm going to start by looking at the endocrine. So the endocrine is important because it's basically the part of the pancreas that's going to release hormones. What's the most important hormone your pancreas is going to produce? Insulin, which is again regulating your blood glucose. And again, I'm going to keep talking about this in many senses because glucose is such an important component for our body. Now, what is the exocrine function implies? It means it does something that's not related to blood or it doesn't get released or release material into the blood. So in this case, exocrine means that it's released into a cavity, which is your digestive system. So pancreas is actually producing a bunch of digestive enzymes that are going to be responsible for the process of digestion. So basically how you digest your food. So things like lipase, DNase, protease, um, pepsin, trypsin, things like that, right? So this is basically, or amylase, these are exocrine digestive enzyme, or exocrine function of the pancreas, creating these digestive enzymes to simply break down your macromolecules that you have consumed to smaller pieces. Now, when we're talking about gene expression, again, the whole idea of this epigenetic is creating different levels of expression of the genes. So differential gene expression produces either an endocrine cells or exocrine cells. So you basically start with the progenitor cells. Those are the ones that haven't differentiated, right? So some progenitor cells are going to follow and transcribe certain factors, and that leads to the exocrine pathway being activated, which means those cells are going to eventually become cells that produce digestive enzymes. Some progenitor cells respond to a different signals, And they are going to yield cells that are going to be part of the endocrine system and therefore produce hormones such as insulin. Here is a depiction of what we're talking about. Again, here is a progenitor, here's a stem cell that has the ability to replicate, right? So it's replicating itself. And then it also has the progenitor cells right here, where it hasn't really determined if it wants to be an exocrine and endocrine, right? So these arrows basically describe the factors, any type of factors, external and internal, can target some of the cells to be exocrine and eventually release, become an exocrine cells, and therefore release digestive enzymes. Or they can be endocrine, and there's different type of them, but we already talked about cells that produce insulin. They can produce glucagon, which is opposite of insulin. So this one decreases blood glucose. Sorry, I can't spell. Glucose, and then this one is going to help you increase the blood glucose. So like when you're fasting. You also have hormone called somatostatin and pancreatic polypeptides. I don't really necessarily want to talk about them, but again, these are hormones that are being released through the endocrine cells that have been created based on the expression of the genes and the cofactors. Now, again, we're talking about epigenetics and what are the mechanisms that allow us to control our gene expression. And I'm going to keep reminding you, gene expression basically means protein synthesis, which proteins were going to be produced based on the epigenetic nature of epigenetics, basically. So where these controls come from? One of the most important controls is the chromatin remodeling. And again, if you guys remember, chromatin was basically a level of condensation for the DNA where you have individual nucleosomes, which is basically DNA wrapped around histones, and then you kind of condense them a little bit more. So right here, these little circles basically identifying your histones, right? And if you kind of look at this structure by itself, not anything else, that was your nucleosome. And if you're really starting to pack it, which is forming something like this, that would be your chromatin. And then this is your chromosome. So chromatin remodeling is basically affecting this part and how histones are wrapping the DNA around them. Another thing we're going to spend some time on is microRNA. Now, it is important to note the following. Chromatin remodeling basically is like an on and off switch, which means if it is present, you're either activating a gene, allowing it to be expressed, or you're turning off a gene or inhibiting a gene, so which means there wouldn't be any gene expression or protein synthesis. MicroRNA is more affecting the level that's present in the cell, so it may slow down the production or increase the production, but it doesn't necessarily stop it. So let's take a look at this, chromatin remodeling. So there are components that can help you, again, control the gene expression. And this is basically looking at how the histones have wrapped themselves, how is the DNA wrapped itself around the histones. So when we're looking at chromatin remodeling, there are parts of the DNA that will be exposed because of the way it has been wrapped around the histone. And there are parts of that DNA that's going to be basically not reachable or unreachable because it is wrapped around the histone and it's not accessible for the process of transcription. Now, as I mentioned, there are components that can allow this process to be turned on, which means you're going from no transcription to a full-blown transcription. And there are scenarios that basically allow this gene to be turned off or allowing this expression to be turned off. So you're going from an on to an off position. Now, what are the main ways or main molecules, more specifically, that can interact with histones and allow the process to basically start. Or in some cases, you may even remove a molecule that allow the interactions of it with histones change the transcription from the off position, quote unquote, to an on position. So the first thing that I want to describe here is acetyls. Acetyls are basically a group of molecules. We're not going to go into the chemistry of it. Here is a kind of an example of what acetyls look like. And notice that it says if you're adding acetyl to your genetic information, that will lead for the transcription to go into the on position. Same results would be seen that if I add phosphates to this process or to my gene, that also triggers an activation of transcription. I will also see the same result if I remove some methyls, which is CH3, from my DNA sequence, and that also act as a transcription on signaling. So 2 is an addition of something, 1 is removal of something that allow this process to be initiated. Here is an example of what we have described. I said that histones, these purples are your histones, or these kind of circles. And notice that they were part of the DNA that is tightly wrapped around the histones, and that makes those portions of the DNA not accessible. So they would be hard to read, and they would be basically hidden. But if I'm adding or going through the process of acetylase, addition of acetylase or phosphate, or removal of methyl, what I can do is expose parts of the DNA that I want to transcribe. And that makes it easy for the RNA polymerase to read the DNA and produce your mRNA through the process of transcription and eventually express your gene in the form of a protein. So this is very important for us to, again, making sections of the chromatin or DNA available as it has wrapped itself around the chromatin. Wrapped itself around histones. So we talked about acetylated or acetyl groups that are added, and that allows the transcription process to basically be initiated and sped up. So how does it do that? So imagine this is your histone, that purple, again, this whole thing is your histone. Kind of look like an alien with 4 arms, right? Histone tail, it says. And right here, we have this structure right here, so it's called Tata box. If this is something we'll talk about later, but Tata box is very important because it's kind of like a signal or a promoter that allows the RNA polymerase, which is this biggest structure right here, to come in and bind to your DNA for the process of transcription. So on the top picture, we see that Tata box is basically on a region of the DNA that is inaccessible to the factors that can turn on the transcription. At the bottom, you can see this basically protein complex come in, right? That kind of shifts the histone, that moves the histone, which makes the Tata box available. And as a result, you are turning on the process of transcription by allowing the RNA polymerase to attach to it. So that's how acetylated works. Same aspects also applies to if we're looking at addition of phosphate or removal of methyls. Now, I wanted to kind of show you this picture. I think is really cool. Again, looking at the levels on the top, we have the chromosome. We have unraveling of the chromosome to form chromatin. And then lastly, if you super open it, you will see your nucleosomes, which is basically DNA wrapped around the histones. So notice here on this side, we have basically a gene that needs to be expressed, right? But the DNA for that gene is not available, which means gene is inactive. Therefore, it cannot be used for protein synthesis. Now, if you have any type of epigenetics components, that epigenetic factor can come in interact with the histone and make that section that was before inaccessible, so right here is inaccessible, making that accessible, which means now that gene can use protein. Therefore, it is an active form of the gene. Now, one of the other ways for us to control the gene expression is looking at the microRNA. The microRNA is one of the coolest things So microRNA, in this case, in this scenario, are targeting the mRNA, and they're very short sequences. They're about 21 to 22 base long. So basically 21 to 20 nucleotides put together. And remember, your mRNA or your genes can be thousands of nucleotide long. So this is a very small sequence, and you can see it if this is your mRNA. The microRNA or miRNAs are relatively small. So microRNA can control the gene expression, which again, protein synthesis, by doing two things. One of the things that you notice is that the microRNA can attach itself to mRNA inside the cytoplasm of the cell, where basically the translation is going to take place, and it will stop the translation from taking place. How does it do that? One way that it can do it is mark that mRNA for destruction. So basically it says this mRNA is not needed, take it away and pull it apart and degrade it. The other way is it can temporarily preserve the mRNA and say, I'm not ready to translate you yet, therefore we're going to stop the translation and start it later. For those who might not remember, I'm just going to show you this in that sense of the process. So DNA became mRNA, right? This is the process of transcription. And then mRNA was used to make your proteins, which is the process of translation. So when we're looking at this process specifically, the microRNA is actually targeting this portion of the process by interacting with the mRNA. But for the previous scenario, when we're talking about your acetylation, we're actually focusing on this portion of the process where DNA needs to be available so we can transcribe it and eventually undergo the process of translation. Another thing we need to know about microRNA, this is actually kind of a common sense, but we want to kind of walk through it. So microRNAs can be underexpressed. Again, underexpressed means you have very low level of them, abnormally low levels. So let's say if this gene is controlling a protein that normally is supposed to be in a certain level, if I don't have enough of that microRNA, then that gene level is not going to be controlled, and you're going to have really high amount of that gene being expressed. More specifically, when I use the word overexpress. So if you have low miRNA, that usually translates to a high levels of gene expression, which is again makes sense because there is no monitoring or ways to inhibit the process. Vice versa is also true. If you have really high or overexpressed microRNA, so you have really high levels of miRNA, that means you're suppressing the process of gene expression, which means you're inhibiting or you under expressing that gene expression. And again, gene expression means low proteins. And again, kind of think about it. If you have too much of a protein or too little of a protein, it can have significant effect on the cells, tissues, organs, and you as a whole. Now, why do we care about microRNAs? Why is this so important for us? It actually can be used to treat cancer. which is, if you kind of think about many of the progresses in the science nowadays, it's the goal is to, at least medical science, I should say, it's to basically eliminate or treat cancer. So let's say if you have a cancer cell that has microRNA genes in them, and those genes are basically damaged or reduced by mutation, which means those microRNAs cannot do what they're supposed to do, right? So it means the level of, oh, I'm sorry, guys. That means the level of your microRNA is, the level of your microRNA is really low. Again, remember that inverse relationship. If my microRNA is low, that means there is something that's being overexpressed. So, so if you have low levels of microRNA, which is what I have here, it can causes what? Overexpression of the genes. And as you can see, cancer is usually something being overexpressed, which is not something we want to do under normal condition. So that high increase in the production of proteins can lead to cancer. And this is starting, the reason that it happens, the reason we have high levels of protein production is because there is a decrease in our microRNA that was basically controlling the levels of that protein. Now, methylation. Methylation simply means addition of a CH3 group or methylene group to a sequence of DNA. So first of all, I know this is a DNA because it is a double helix or double-stranded DNA. So if you add a methylation to your DNA sequence, which is identified as one of the common epigenetics changes that we can expect, it actually inhibits the expression of certain genes. So basically, by adding these groups here, you are stopping the process of what? Gene expression, which means you do not produce any protein. So you're basically blocking your DNA from being transcribed and eventually translated into the protein. The methylation process might stop tumor-causing genes from turning on, which preventing cancer. Which again, if we're looking at it in terms of epigenetics, right? So if you have certain characteristic in your environment, you can actually have this methylation take place. And when that happens, it means you're turning off genes that can lead to cancer and therefore protect yourself against cancer. Can you imagine if you can find those kind of DNA methylations that can affect a specific gene in somebody who has cancer? It means that we literally stop the cancer on its place. They can't go any further because there's no gene expression. Now, if you ever wonder, we keep talking about genes and we talk about the fact that genes encode or lead to gene expressions of thousands of different proteins. Now, if we look at the whole DNA sequence that we have inside ourselves, look at these numbers, it's staggering. Only 1.5% of all of our genetic information actually encodes for what? Proteins. Which means if you have 1,000 miles of DNA, and this is arbitrary numbers, only a mile and a half of that genetic DNA is going to encode for those thousands of proteins, and that's it. Which begs the question, what is the other 98.5% of your genome? Really cool fact. You actually have viral DNA that's embedded as part of your genome, or your DNA, I should say, your sequences. There is a lot of non-coding RNA. There are introns, which is part of the mRNA before it is getting ready for translation. You have things like promoters, which we're going to talk about in more detail later on, and as well repeated sequences, which accounts for a good chunk of your genetic information. So most of the genomes are transcribed, right? Which means transcribed means you're going from, again, DNA to an mRNA. But just because you have transcribed something, that doesn't mean it's going to lead to what? A protein. So you may go the first part of the process, which is the transcription, but that does not lead to a synthesis of a protein. So those DNA sequences in this case are referred to as basically, well, they're not referred to as, what I should say. They're not technically junk, but we either don't understand what their function is, or there is no function that it might be associated with, I'm sorry, it might be associated with how other genes may be expressed. So one transcription or part of the genetic information controls the expression of other genes. This is also a very interesting fact. If you sequence your whole genetic information, 8% of that genome is going to be belonging to RNA viruses. Now, if I say this, everybody gets kind of freaked out, say, wow, so does that mean that I'm going to be continuously sick because I'm carrying 8% of the viral genetic information or genetic information that came from viruses? And the answer is no. These are actually signs or evidence that you have had infection. And as you get older, basically because you undergo more infection, then that percentage of the viral DNA increases over time. It's also important to note that the genetic material in the human chromosome is also called endogenous retrovirus, or HERV, or HERVs. And these HERVs have basically have exchange parts, which means they have had basically a DNA recombination. Either parts have been changed, edited, or added. And these viral genetics, you should not really worry about them because there's so much mutation and changes that has happened to these DNAs that they can't really harm us anymore. They're just literally part of you, who you are. Now, many people, which I think this is really interesting, if you have certain type of medical conditions like MAL, MS, or melanoma, which is a skin cancer, you will have an overexpression of these HERVs or HERVs, which means that even though these viral chromosomes may seem harmless, but they may have a contributing role in terms of the cancer development, but we're not sure yet. Now, non-coding RNA. This is a good one too. Non-coding RNA, just means that we have the whole genome being, again, undergoing the transcription, which means the DNA is becoming RNA. But most of that genetic information is not coding for any proteins. And therefore, we call them non-coding or ncRNA. What does that include? So it includes all the RNAs other than mRNA, because mRNA, as soon as you say mRNA, that means what? It's encoding for proteins. So we're saying anything that basically gets transcribed but doesn't get used for protein synthesis. So what does that include? It includes your tRNAs. tRNAs are, and this is something we'll talk about later, are used in the process of translation. rRNAs are also important. These are the ones that are used to synthesize ribosomes and other factors for the purpose of protein synthesis. And in reality, again, there are hundreds or thousands of non-coding sequences that are present in our body. And a lot of them might be transcribing to what we call pseudogene. So pseudo implies means fake, right? So even though they look like a gene, they do not encode for a protein. They basically do not produce a protein. They're not translated into proteins. Now, another part of your genetic information is dedicated to what we call repeats. So, repeats are basically these highly repetitive, continuous sequences that can be shown all over the genome. And again, there are, in reality, they're referred to as transposons. And guys look at, and I'm going to show you this and I'll come back to this. Transposons, which is what we're going to talk about, those repeats are about 45% of your genetic information. So what are these transposons? So again, these transposons are basically repeated sequences that can be found throughout your genome. And sometimes what they do is they either create a brand new gene or reversing mutations. Sorry, I should say new genes. They create mutations in an existing gene or reversing the mutation that was already existing within a gene. So they basically could have kind of a positive effect or a negative effect. We're not going to go into that details though. So what I wanted to show you in this picture is simple sequence repeats, which basically means we're looking at a very short sequence like the AGAT sequence that can be repeated throughout the genome. So we have 3 different people here, and we have the same repeats in 2 different locations. The first site of these short, what we call short tandem repeats or STRs, the first person has 7 repeats in the first position and 6 repeats on the second one. Our second person has 4 repeats for the first position and 19 for the third one. Sorry, for the second one. And then again, 12 and 14 respectively for our third person. So these are very unique to the individuals, and they do get passed down from parents to the children or their offspring, and they can be used as a fingerprint. So basically see how that person might be related to the parents or to other people. Now, I briefly mentioned pseudogenes, which basically means they don't get translated. They used to probably be genes, but they're not genes anymore. So the segments of the DNA that are structurally, again, resembling genes, but they do not encode for a protein. In many cases, they believe that this is what used to be. I'm going to repeat this. They used to be encoding for a gene possibly, but because of their mutations or changes that has happened to it, they do not encode for proteins anymore. So we can refer to those as genomic fossils, or what we are treating them as basically junk DNA, something that there's no point of keeping it, or we're not really doing anything with it. Now, it has been recognized that some of the pseudogenes, I'm going to be specific, guys, not all of them, most of them, they're basically identified as junk. But some of these pseudogenes do have an ability to control their parent genes, so the genes that are similar to the pseudogenes but encode for a protein. So they can affect the parent genes and control its regulate its expression. So basically, how much of the parent gene basically is produced may be dependent of the specific pseudogenes. We don't necessarily have a lot of information about that. Here's again the picture that I wanted to show you, kind of highlighting how insignificant and at the same time significant the genome is, and how important it is that even though only 1.5% of your whole genome encodes for our I guess coding sequences, they're still very, very important for us. So this is the one that we're talking about. This is the one we're really concerned about. And I want to finish it up by looking at a couple of charts. I'm not going to go through all of them. The goal of this is for you guys to look through it, get an idea of what are these, again, that 98.5% of the genome, what they're actually do inside our body. So again, we talked about some of this. We didn't need to talk about every single one, but we talked about the viral DNA, the non-coding sequence of tRNA and RNA, long non-coding sequence, pseudogenes. Introns I didn't talk about in details, but we have mentioned it when we talked about introduction to the genome, so it should be familiar to you. Promoter sequences are very important. They're used for both the DNA replication, transcription, and translation, so their presence must be essential to those three functions. MicroRNA we talked about in details, including transposons. Centromeres are the regions where the chromosomes are connecting, and then telomeres are the ends of the genetic information. And then I didn't really talk about segmental duplication and simple sequences, but if you want to learn about them, you're welcome to do your own research. And that's about it for our lecture today. It's kind of on the short and sweet side, if you want to think that way. But anyway, thank you for listening to the lecture, and let me know if you do have any questions. We can, of course, meet, and make sure you keep up with your studies.